A very good day to my fellow doctors, medical students, friends, and my beloved audience. My name is HARRINESHKA NADARAJAH, a first-year medical student in University Malaysia Sabah. I would like to convey my heartfelt gratitude to Professor Dr. Zainal Arifin for giving me this opportunity to share my knowledge and interest on the brain. It is truly undeniable that the basics of medical science is anatomy. So let's delve into the anatomy of the brain, neuron anatomy, and assisting cells in the nervous system briefly. The brain consists of three main parts: cerebrum, cerebellum, and brainstem. The cerebrum is organized into two hemispheres that are connected by a large bundle of white matter tissue called corpus callosum. Each hemisphere of the cerebrum contains 5 lobes: frontal, temporal, parietal, occipital, and insular. These portions together are called operculum, which means covering of the insula. The outer surface of the cerebrum exhibits many elevated ridges of tissues called gyra that are separated by grooves called sulci. The gyra and sulci increase the surface area of the cerebrum, providing it with its characteristics of convoluted appearance. The brainstem is the most subtle part of the brain. It is made up of midbrain, pons, and the medulla oblongata, each of which have their own structural and functional organization. Okay, so that's it with the anatomy of the brain. Now let's go into the microscopic view and look at the neuron. We all know that neuron has nucleus, which has a cell body. The nucleus is the control center, and soma is a structure or cell body that provides energy and support for the neurons. Okay, so branching out of the cell body, we have dendrites, which receive signals from other neurons and passes through the cell body. Okay, next we have axon, a long thin structure that carries electrical signals away from the cell body. Covering the axon, we have the myelin sheet, which is formed by Schwann cells for insulation and faster signal transmissions. And talking about fast, we also have nodes of Ranvier, which is in between two Schwann cells, which helps in saltatory conduction. Okay, at the end of the neuron, we have axon terminals that connects one neuron to another, or one neuron to another neuron, muscle, or gland, transmitting signals throughout the chemical messengers called neurotransmitters. Okay, so that is it for neuron anatomy. So we have 3 types of neurons: sensory neuron, motor neuron, and interneuron. And we also have assisting cells in the nervous system, which helps in maintaining the neuron, but we will specifically talk about neuroglial cells because it will be related to the disease that we will be talking about later. Now that we have talked about the anatomy of the brain and the neurons, let's delve into the physiology of the brain. Okay, speaking about physiology, all I can think about is action potentials that happens in the nervous system. Okay, so first we have the resting potential in the action potential, which means there's a difference in charge inside of the membrane, which is more negative compared to the outside due to the sodium and potassium ions. So this is the resting potential. So when a stimuli triggers is strong enough, it triggers the threshold, which opens the voltage-gated sodium channels, and sodium ions rush into the cell, making the inside more positive. This phase is called depolarization. The positive charge travels along the axon like a wave to keep it moving efficiently, and the signal jumps between nodes of Ranvier along the myelin sheath, speeding up the transmission. Okay. Next, we have repolarization. The potassium channels open and the potassium ions flow out of the cell, restoring the negative charge inside. And then we have the potential returning to resting potential, which means the neurons reset using sodium-potassium pump, which restores the ion balance, bringing the neuron back to its resting potential. Okay, so yeah, that's all for action potentials. Now let's look at the chemical reactions that are involved in the central nervous system. Okay, so talking about chemical reaction, the most important thing that comes to my mind is synapses. Okay, synapses in layman terms is how neurons communicate with each other. So yeah, the process begins at the axon terminal of the sending neuron, also called the presynaptic neuron. Here, the electrical signals trigger tiny sacs called vesicles to release neurotransmitters. So, the neurotransmitters released into the synaptic gap, so these neurotransmitters are released into the synaptic gap called synaptic cleft. Okay, on the other side, the receiving neuron or postsynaptic neuron has specialized structures called receptors on its dendrites. These receptors are like locks and neurotransmitters act like keys. Okay, so when neurotransmitters bind to the receptors, they trigger a new electrical signal in receiving the neuron, continuing the message. So this is where one of the characteristics of synapses is once the message is sent, the neurotransmitters are either broken down or reabsorbed in a process called reuptake or diffuse away to ensure clear communication. So synapses are key to brain function, allowing neurons to work together to control thoughts, movements, and everything in between. Okay, so now that we have mastered the basic of anatomy, histology, physiology, and also the chemical reactions, let's put on our white coats and assess some clinical cases. Okay, a little drawing that I made for better explanation. Let's talk about the clinical case. I would like to focus mainly on Alzheimer's disease, in particular because it is a very severe form of neurodegenerative disease, which is closely related to the brain. So let's look at the symptoms, causes, risks, and treatments of Alzheimer's. Alzheimer's disease is a progressive brain disorder that disrupts memory, thinking, and behavior. At its core lies the accumulation of toxic proteins called amyloid beta plaques. So these amyloid beta plaques are cleared away, but in normal healthy brains. But in Alzheimer's, the fragments of this protein build up between neurons, forming plaques. So these plaques interfere in the communication between brain cells and trigger cellular damage. So remember that I mentioned about the neuroglia, right? So these neuroglia are assisting cells in the brain, which helps to clear the debris and maintain a neuron. But in the case of Alzheimer's, neuroglia becomes overactivated by the growing amyloid plaques. So while they attempt to clear the plaque, this constant activation within themselves causes inflammation, which further damages the neuron. So this becomes a vicious cycle where the amyloid beta plaques build up and the microglia overreact and the neurons are harmed further, leading to uncontrollable neurodegenerative disorder. Okay, so the good thing here is we have few treatments that are in the direction of improving maybe not entirely curing, but helps to reverse the adverse effects of Alzheimer's. So one of the treatments are stem cell therapies. So in stem cell therapies, we have to think of the brain as like a broken power plant, and the stem cells help to work as like technicians to repair the brain. So stem cells can transform into new functional neurons to replace the broken ones. They also help to repair connections, synapses, between neurons to restore traffic flow and the brain's communication network. The stem cells also release chemicals that reduce inflammation, clear debris like amyloid plaques, and help the brain to run smoother. Okay, yeah, so that is all about brain and the disease related to it. But last but not the least, how can I end this discussion without talking about the doctor that inspired me throughout my journey and still continues to, Dr. Nur Amalina Bakri, which is a renowned Malaysian female neurosurgeon celebrated for her significant contribution to the field of surgery and medical research. So we can see that Dr. Nur Amalina Bakri lived a life of excellence and advocacy from SPM, having 17 A1s, being awarded prestigious scholarships, and continuing her A-levels with also 5 straight A's, and then now currently working in London. It all really inspires me and gives me hope that anyone can do it with hard work. Okay, so yeah, that is it, everyone. Thank you so much for sticking around until the end. Have a nice day, and I hope all your dreams will come true. Thank you very much.